every bioreactor run that has started needs to start with a smaller culture first, and then you usually scale it from a vial to a small shake flask to a bigger one, and then you inoculate your bioreactor. So shake flasks are really used diversely within the microbial space. And in cell culture, it's more that we are using single-use flasks because of sterility things. I mean, Corning flasks, I think everybody knows them. And they're really more used in the clone selection part.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. Should you be using shake flasks, or is it better to move into a more elaborate small-scale bioreactor system? Today we're talking exactly about that with Jens Bayer, who is a deep tech entrepreneur and experienced commercial leader, and he currently serves as the Chief Commercial Officer at Scientific Bioprocessing. He also co-founded Aquilo Biolabs, and under his leadership, the company experienced rapid growth and a successful exit through acquisition by Scientific Bioprocessing. And we are talking about the advantages of shake flasks and how potentially you could transform a shake flask into a bioreactor. Stay tuned. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Hey Jens, welcome to the Smart Biotech Scientist podcast.
Hi David, nice to be your guest today. Thanks for the invitation.
Absolutely, it's a pleasure having you on today. Jens, share something that you believe about bioprocess development that most people disagree with.
Wow, that's a challenging one for the beginning. Okay, let's provoke. It's early in the morning. So I would say shake flask can play a pivotal role in bioprocess development, and it is possible to do scale up from shake flask to later stages. That is my opinion, and I think some people would disagree.
Yes, I agree. Some people will disagree, and I'm happy to talk about that, exactly that. But before we do that, let's talk about yourself for a moment, Jens, because your background is quite diverse. You're blending science, business, and entrepreneurship. So I'm curious, how did you find your way into the world of bioprocessing, and what sparked your interest in this field?
So maybe let's start at the beginning. I studied biotechnology at the RWTH Aachen, so one of Germany's leading technical universities. And in Aachen, biotechnology is really classically meant, so it's not just a molecular biotechnology, but also, let's say, more the engineering part of things from upstream and downstream. So we had lectures on these topics, and I thought it was quite interesting. And then there's one professor in Aachen, Professor Büchs, that was leading the bioprocess engineering part, and he's really a key opinion leader within the area, especially around shake flask, co-developer of the biolector system, around the Ramos technology. So there was very early on in my bachelor studies this upstream bioprocessing focus and fermentation and microbial space. And that's really, I think, where it originally came from. And then, of course, later, when David and Konrad, two of my friends from the bachelor program, asked me to join a company they were founding, this company, Aquilo Biolabs, was in the space of upstream bioprocessing and especially sensors. And I mean, I've lived and worked in this industry for the last 10 years. And then you talk to customers and clients in this field, you talk to the shaker and bioreactor manufacturers. I'm living the upstream bioprocessing part since 10 years, and that's really how I got into the field.
Well, exciting. And since you've seen different parts of the industry, based on your experience, what do you think are the main challenges biotech scientists oftenly face in development and the manufacturing of biologics?
I think I mentioned one of the challenges directly. Bioprocess development is a mixture of biology and engineering. So you kind of have to master two disciplines. That takes the right education, but it also takes the right type of person that can actually do this. And by itself, it's just a very complicated field. You just don't need to be good at one thing, but you need a team that's usually multidisciplinary. And then, of course, we all know the biggest challenge it's one thing to develop maybe an idea or have a proof of concept or a strain or clone that can do one thing. But we all know that a functional commercial process where you really reliably can produce a product of high quality with the right titers at the cost structure that you had planned is a completely different animal. And that's, of course, the scale-up challenge that many people talk about. And then over the last years, we've seen it. Budgets and costs are getting more and more important and more tighter controlled. And timelines, expectations of time to market has definitely also not decreased, but people want to see these things faster and faster. And so if you take something that's very challenging by itself and then you add cost pressure and time pressure to it, then it doesn't get much easier, right? So that's, I would say, what key areas of challenges are for people.
Now, let's dive into the main topic of today, the shake flask. And it's undoubtedly a fundamental tool in both microbial and cell culture bioprocess development. Many people have been using it and are still using it. Where probably some people will not agree is how much we should be using it in the future. So I'm really excited about this conversation. Could you elaborate on its current role? So both in the industrial and also, I'd say, in the research area, and highlight any significant advancements it has facilitated?
It's a complex question, but I will do my best. So first thing I want to note, when we founded the company, that was 2014, there were already investors that told us, no, we're not going to invest because the shake flask is dead. And you won't find it in the next 10 to 20 years. And now we are here today, and the shake flask is as strong as it has ever been. So I'm very convinced that you won't get rid of it. The reason is it is so easy to use. It is one of the most parallelizable bioreactors out there. It's low cost and it has a very small footprint. In its nature, it's going to be very difficult to take it away. Of course, the problems around it are you kind of operate in a black box. If you compare it to a bioreactor, you don't have all these sensors. You don't get DO data, pH data. You have no automation or control for something like fed batch or stirrer speed. That's why it has its challenges. But the role that it plays today, we have to look at two different market segments, I would say. One is the microbial space. So that's what we define as, let's say, bacteria, yeast, filamentous organisms, maybe even plant cells and microalgae. And then on the other, we have more the mammalian cell culture space. So let's say CHO, HEK cells, insect cell lines. In the microbial world, the shake flasks, we're talking about glass shake flasks that are used, then are autoclaved, and they are really used for applications like strain screening and selection, like media optimization, even scale-up experiments, but especially seed trains, right? So for example, every bioreactor run that has started needs to start with a smaller culture first, and then you usually scale it from a vial to a small shake flask to a bigger one, and then you inoculate your bioreactor. So shake flasks are really used diversely within the microbial space. And in cell culture, it's more that we are using single-use flasks because of sterility things. I mean, Corning flasks, I think everybody knows them. And they're really more used in the clone selection part. So let's say you are done in the clone development part and high throughput screening, and you have, let's say, 100 plus candidates now, and now you're trying to really narrow it down to your top 5, top 10 clones that you can then actually maybe use in an Ember system and so on. So I would say that's the role the shake flask plays, very diverse. And from the smallest startups to the biggest pharma companies, regardless of budget, everybody is using them. So they are everywhere. That was the one part of the question. You asked for recent advancements. I would say we as Aquila Biolabs helped quite a lot in that space to bring more data to the shake flask. So there are now sensors for biomass, for pH, for DO. There is now feeding technologies. There's also a couple of other startups in the space that have, let's say, evolved over the last 1-2 years. So I think people are really taking this commonly used shake flask and say, we need to digitize this more. We need to add tools to it in order to improve it. And I think it's really time on the urge to become smart shake flask and play a completely different role than what it has until today.
So what I'm hearing, Jens, The main advantage of shake flask is that they're simple to use and they're quite cheap to use, right?
That's absolutely correct. Imagine like this, right? Imagine you have a shaker there, and I tell somebody, can you please run 6 runs in parallel on this shaker? You take 6 flasks, you go on the sterile hood, you pipe it, everything together. To be challenging, my dad is a sports commentator, but I think if I gave him 1 or 2 hours of training, I think he could put this together. If I told my dad, hey, can you please fix these 6 bioreactors here right now and run them and, control stir speed and choose the right impeller type and so on, there's just no chance. And I think that's exactly the thing. Biologists tend to keep it easy and go more towards the safe bet, and that's often the shake flask. And I think many people are a little bit scared of bioreactors because it really screams engineering at you when you look at them, while a shake flask screams like, hey, I can handle this. This is easy, right? Put it in there, I get my data. But that's also one of the biggest disadvantages of shake flask, because it is a small bioreactor and people should treat it more like this, but they just don't. They don't know enough really about what they're doing in shake flask.
So besides the black box, let's talk about other disadvantages or challenges. What have you observed?
I would like to highlight the black box challenge because I think it is a big one. So today it looks like this. You're putting it in there, and in the bioreactor you're getting pH and DO data in real time, right? Like you don't get anything from a shake flask if you don't sample. But we know that sampling is not good, because the moment you stop shaking, even if you just sample from one flask, all of the flasks on the tray essentially stop getting oxygen in and your cells stop breathing, right? It has a direct effect on the metabolism. If you do this several times, it's just going to not lead to consistent results, and it's not going to lead to optimal results. So people may say, we don't have a data challenge, right? I can just sample. Yeah, but the sampling is not good for your bioprocess. It's literally like you would always stop your bioreactor. Nobody would do that, right? You wouldn't go there, stop your bioreactor to take a sample and then continue stirring. Nobody would do that. But in a shaker, that's what people do. Of course, you have things like feeding, like run fed batch, right? You're limited to batch really in shake flask, but later processes are often operated under fed batch conditions. That's a big one. And then, of course, you have oxygen transfer, right? Like in a bioreactor, you can control this by stir speed, gassing rates, gassing mix, and all of this. But in a shake flask, you're really limited there, or people at least don't know about the options that you have to control this a little bit. I want to highlight the, in my opinion, biggest challenge of them all is also what I mentioned. People don't treat it like a bioreactor. They simply look at the biology, put my cells in, put my media in, and I forget about it. They don't have the mindset of, now I'm running a small fermentation. Now I should be really thinking of, what do my cells need? How do I optimize this so that I get the maximum titer, that I get optimal results, and that it becomes reproducible? And I think, so I would highlight that shake flask has a challenge of awareness and knowledge that people need to read up a little bit more, because it's such a niche topic and nobody really knows about this. And that's the advantage we had with Professor Büchs in Aachen, that you early on have somebody that tells you about it, and then you're quite surprised. I'm in this space for 10 years, and I've seen the biggest pharma companies and smaller startups, and you don't meet super many people that know exactly what's going on in that space. And that's interesting to me.
When I was working in technology innovation, we would have a lot of discussions about what is the right tool to use. Should we have a shake flask? Should we use spin tube? Should we use an Amber 15? Should we use wave back? So we have now a lot of different options at our disposal. And you mentioned the black box, and I think certain systems are better because you can sample much more easily. You get a lot more information out of that. Others have the drawback that they're maybe too complicated, and shake flasks are much simpler. Can you just tell us what are a bit the differences in, like, when would a shake flask make sense, and maybe in certain situations it would probably make sense to go into an Amber system or a wave back system? Like, what are your thoughts about that?
I would say the most important thing for mine is to say, I'm not standing here and saying the shake flask is the solution for everybody. It's a very complex world out there. It depends on very many factors which toolbox you should use. So, for example, are you working in cell culture or in the microbial space? Are you dealing with an aerobic organism or an anaerobic one? Are you dealing with a bacteria or a filamentous fungi? Am I a big pharma company with a very advanced team, lots of expertise? We've done this many times. Or am I a small SynBio startup, handful of people, and we're doing this for the first time, right? All of these kind of factors influence the toolkit that you can afford and maybe want to use. So there is not one. I would say the standard we see in industry in the microbial is starting in microtiter plates, maybe even working with something like BioLector, and then moving from there to shake flask level to understand your process or your strain a little bit deeper. Also using technologies like the CGQ for automated biomass monitoring, like LIS for fed batch experiments in shake flask, and then moving to the benchtop bioreactor sites, maybe more for scale-up experiments. In cell culture, it's more, you start in multi-titer plate as well, but there we definitely see the Ember system then way more. It's kind of become the gold standard, I would say, and it's just because it works. It's a very great technology in my opinion, but of course it has its downsides. Like you need people to know how to run it. You need to have the budget to afford the OPEX and running costs for it, but it's a technology that works. And so you can see how all these different factors come together to choose the right thing. You asked me, when is the shake flask a good solution? I would say everybody should use it for specific experiments where the bioreactor is too expensive. Like if you try to understand your organism better, if you, for example, do some media optimization, strain screening experiments, early testing to just understand it, everybody should use the shake flask. For somebody who maybe even does scale up there, I would definitely say if you don't have the funds right now to afford all the fancy tech, it can be done in shake flask. You can scale up from shake flask. You will need to read a lot about it and you will need to know what it does, what you do, but there is ways to definitely make it happen. And that's when I would recommend more the shake flask.
So that means that even though you're using a shake flask, you can make sure that you're able to get comparable process at the larger scale, or you think you need to put a lot more work into that in order to have something then that's consistent and robust at larger scale?
I wouldn't say you can do it for every product and every strain and organism you're working with. There will be examples where it just doesn't work, but there are countless examples, and you can take publications from Professor Büchs where you can see that you can scale processes from the shake flask level also to benchtop reactors and beyond. So it depends on your experiences and maybe also with which tech you're comfortable, but it can be done for sure.
Hey, smart biotech scientists. Have you been enjoying the conversation with Jens Bayer? Jens Bayer is the Chief Commercial Officer at Scientific Bioprocessing. And in part 2 of our conversation, we are going to dive into the use of sensors. So stay tuned for an exciting conversation this Thursday. And before that, I am curious, what do you think about today's conversation? Please tell us by leaving a review. And perhaps you're not so sure whether you should use shake flasks, whether you should use an Amber system, whether you should use some small-scale benchtop bioreactor system, or whatever it might be, whatever question you might have. I want to help you get started and to get clarity. So I'm offering you a free 30-minute assessment. Just book the call using the link down in the show notes, and I'm looking forward to seeing you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.